All right, cognitive neuroscience class here. We're going to talk about brain imaging technology so that you understand a little bit more about the technologies that you're reading about. So there's some common issues that apply to all of the brain imaging technologies that are used. And the first issue concerns resolution. There's two types of resolution. One resolution is called spatial resolution, like three-dimensional space. So this is our ability to identify particular structures, that the signal is coming from a particular structure. So I'll give you an example here. You're familiar with this concept, even though you may not know it as spatial resolution. Giving you an example of this, you have a high resolution picture of our atrium before it was painted, and then here I decrease the resolution, so there's just fewer pixels in this particular image down here at the bottom, and so you can't see the structures as clearly, right? So here in the upper image you can make out that it's clearly some trees and maybe a trash can, and here you just sort of see it as a blob. So that gives you an example of how we can sort of have technology that gives real clear pictures of brain structures and some technology that doesn't give clear pictures. So that's spatial resolution. Now, temporal resolution, of course, our structures can be differentially active. They can be active at different points in time. So temporal resolution refers to its activity changes over time. So here's a good example of an FMRI, and you can see that both the left and the right motor cortex are active, but the question is, which one was active first? Was the left active before the right? Was the right active before the left? Or were they active simultaneously? We need some additional information to come to that conclusion. Now, another common issue is we can't just ask people to turn off their brains and turn on their brains. They're constantly working, even when you ask them to focus on a particular task. So what we do instead is we repeat trials and then we average to extract the signal from the noise. So what you see here on this slide is EEG, of course, and it's showing you what one trial would look like, or if you took two trials of the same cognition and average them together. And you can see that once we get up to like 40 trials or so, a clear signal emerges. So all the random stuff gets canceled out. So you can see like in the early part of this and the later part of this, it's pretty flat because it's random brain activity on those trials, and that gets canceled out. So all of our imaging technology basically uses this averaging principle. Now that means that technology is not going to be great for studying some phenomena that occur infrequently are not going to be suitable for imaging. So you can't just take anything and put people in a scanner and make some good conclusions. There has to be this ability to average across conditions. So think about, for example, like if we were studying autobiographical memory, we would have to have lots of autobiographical memories to average across in order to get a signal. So that could be problematic. You could study it in other ways, but with brain imaging, might not be appropriate. So some techniques are event-related. It'll appear in the name, like we'll talk about event-related potentials. FMRI, sometimes you'll see the word event-related FMRI used, maybe in the title or maybe in the methods section. And what that means is that you can go into the data set and you can identify an individual event, presentation of a particular stimulus, for example, or response, whatever, and that can be isolated. So that means that we can randomize our trial sets and then pick out individual trials that are alike and then average them, because as I was just saying, we use this averaging procedure. So that differs from some other technologies that are not event related, in particular PET, which we'll talk about, images in a block. So that means that everything in a block of trials has to be the same cognition or the same kind of event, because you can't go in and pick out individual events and using that technology. So that means that your trial sequences aren't random in that particular case. Now, for a lot of things, this isn't going to matter. If you're studying movement, things like that, it may not matter. But for things like memory, it matters a lot, because what happens is if you don't have a random trial sequence, people will pick up on the fact that your trials are not random, and that'll change their strategy, and the strategy will influence their brain activity. So that's something to keep in mind depending on what people are studying. Another imaging issue, particularly for fMRI, and PET, is this idea of using the subtraction logic. Now, I will say that this is not as common anymore, but you might see some papers that use this. So this is how it works. So the idea is, if you were to just look at brain activity in a particular condition. So like here in this study, people studied images, pictures, and then took a memory test. And so some of the items on the memory test were old. So you see brain images of when people were looking at old images versus when people were looking at new images. And that's fine, but you'll notice that lots of areas are active. How do we target specific areas of interest, something that's called regions of interest? Well, one way we can do that is subtract. So the idea here is that let's create a control condition that has all of the other forms of cognition except what we're interested in, and then we'll subtract it. So in this particular case, you could do a subtraction between old and new, and that's often done. But in this case, they had two different tests. They had one test where it was a general, just they were identifying whether it was old or new. And another test, they were identifying whether the image was altered or not, whether it was the same. So they changed the size of the image. So is it the same size or a different size? That's what they mean by specific here. And so then what they did is they did the specific minus general contrast and then focused on this area in the left prefrontal cortex that was differentially active. So this is an example of a good baseline. Everything else was the same. They isolated what would be responsible for, the area that would be responsible for making these more specific judgments. But this means that this requires a good baseline. And a lot of times you'll find that research papers don't have a very well thought out baseline. So then they're subtracting and they're getting measures of brain activity or these blobs, but you don't know what they mean. So this is a paper that would be what we call blobology, which means it's sort of the study of blobs, not really the study of meaningful activation. So you really always want to look at their baseline. Did they use a good baseline? If they're using that baseline subtraction technique. Another imaging issue is the fMRI environment is very noisy. We'll see some examples of that, and this can affect some cognition. You can't assume that the environment has no impact on the things that you're studying. And an example of this, Gutchison Park in 2006 did a study where they had young people and older adults, do a basic memory study. And they did it in the laboratory or in the fMRI environment. And they just looked at their performance. And what they found is that they found performance overall. They found a main effect here overall was worse in the MRI environment. But there was an interaction. So this affected older adults more than younger adults. So this is something that you have to assume, right? So if you're just looking at brain image differences between young and older adults, some of it may be due to the environment in which they're being tested. So it's something that's not talked a lot about. It's not mentioned in a lot of papers, but it's something that you should be thinking about as you critically consume the studies that you read. The other thing to keep in the back of your mind is brain imaging evidence is very compelling. And so as evidence of this, there was a study that was done here, and what they did is they had a couple different groups. They had one group who just read a passage that described some behavior with just a table. That's the control group. And then they had another group that got evidence in the form of a bar graph, right? And then they got a third group had evidence in form of these brain imaging slides here, and they just sort of asked them to sort of rate how believable the evidence was that they in this narrative that they were reading. And what you see is if you add a brain image, it gets much more convincing. So that's something to keep in mind as well, that brain imaging is very seductive. So you have to resist that temptation and think critically about it, maybe even more so critically than you would if you're looking at data in another format. Okay, our first technology that we'll talk about is something called computerized tomography or CT scan. It used to be called computerized axial tomography or CAT scan. You may still see it referred to as CAT scan. Axial just referred to a horizontal slice. That was the only view that you could get. Of course, that technology changed. Now you can get any view you want, so they just call it a CT scanner. So the way this works is that you have your patient here or your participant here, and they're lying on a bed, you slide them into the scanner, and it takes an X-ray in a 360-degree arc around their head, and we get pictures of their brain. So this is like an axial slice, a horizontal slice, and you can see various structures in this image. So the strengths of this technology it was the first non-invasive imaging technique. So it was the first time that doctors or scientists could look inside a brain and even actually quantify the size of brain structures without like opening up the person's head in a surgical procedure. And it's still useful for clinical diagnosis. Most hospitals have CT scanners, and if you have a head injury, first thing they're going to order is probably a CT scan so that you can rule out any sort of serious damage to your brain. Now, the weaknesses for our purposes for understanding brain behavior relationships are it only provides information about structural information. So you can look at the size of the structure, see if they're there, but we don't know how they're functioning. So for example, in that image, so in this image back here, this could be an image of a person whose brain is either living or dead. We don't know because we don't get any functional information or temporal information about the activity. So that's a significant weakness. Another significant weakness is the spatial resolution, or ability to see different structures, is relatively poor. To give you a frame of reference for that, the CT can resolve tissue down to 0.5 centimeter or so, whereas the MRI has 1 millimeter resolution. So that means that we can see little cubes of tissue that are 1 millimeter by 1 millimeter by 1 millimeter in size. Okay, the next technology we'll talk about is something called positron emission tomography, or PET. Here's your PET scanner. It works very similar, or looks very similar on the exterior. They all look, actually all these technologies look pretty similar, but you're going to have a situation where your patient, your subject, is lying on a bed, and then they are slid into the scanner, and the scanner is going to take pictures of their brain. So how does this work? So the principle that it uses is it's going to use a radioactive label. So it's called PET. There's also methods that are very similar to this called regional cerebral blood flow, or RCBF. So if you see that name used, very similar. What they're doing is they're injecting a radioactive label into your bloodstream, or maybe you're inhaling a gas like xenon. So there's different labels that can be used. I'll show you a picture of a glucose scan where they take glucose and attach a radioactive label to it. People would like drink a glucose shake, and then it gets broken down and into their bloodstream and absorbed into their brain. This is a much slower scan. This process of digesting the glucose and getting into your brain can take 30 minutes or so. So if you were doing a study like this, you would drink the shake and then wait like 30 minutes or so and then image the person's brain. So it's not useful for lots of things that we talk about, more rapid cognitions, but you might see something. More commonly is making water with a particle accelerator radioactively imbalanced, making it using O15. So O15 will decay. It has about a 90-second decay rate. And so this, no matter what label you're using, the label's absorbed into the blood, and of course the blood makes its way into the brain, and it starts to decay. And when it decays, it will emit a positron, hence the name positron emission. So the positrons, when they collide, as the slide indicates over here, when the positron collides with an electron, it will emit 2 gamma rays. And gamma rays are like a cousin to X-rays. They're part of the electromagnetic spectrum. We can't see gamma rays, but they'll just pass right through your body tissues and can be measured by the PET scanner. In fact, that's what it does. So the gamma rays are perpendicular to the positron, and so the PET scanner itself has all of these gamma ray detectors and is measuring the gamma radiation that's coming from inside the brain, triggered by the radioactive label. So based on that, it creates a map of brain tissue, and essentially you'll have more gamma where there's heavier concentrations of blood, heavier concentrations of label, and so we can measure not only structure but the activity of the brain based on what's called a hemodynamic principle, right? So your neurons, when they're active, they use oxygen and glucose. Well, you need more oxygen and glucose if they're more active. So areas that are more active are going to get more blood flow. So the idea here is we're measuring brain activity, but it's sort of an indirect measure. We're not actually measuring, action potentials by neurons. We're measuring blood flow. Now, that's a pretty good, strong correlation between blood flow and brain activity, but it means that the technology is imaging the brain activity based on blood flow, so the pictures only change as fast as blood can move around in the brain. So we're going to see that that's a limitation of sorts for some methodologies that use that are hemodynamic techniques, I should say. So this is an example of glucose scan. So here you have two patients. So you have one patient, the control patient, and patients who are on cocaine. And you can see that cocaine, the brain is less active in some areas. So that gives you an example of one of those slow scans. Here's a more common PET scan of somebody with tinnitus, and you can see that areas in the temporal cortex are active in this person when they have ringing of the ear. So there is no external stimulus, but yet their auditory cortex is acting like they're actually hearing sounds in their brain. So some strengths of this. This was the first technique that provided both structural information and functional information. So that's really was groundbreaking technology. And even by today's standards, it still has good spatial and temporal resolution for a lot of applications. There's still a couple of PET imagers out there, and so you might see a PET study. You might come across some PET studies. The weaknesses are it's very expensive, so it's basically been replaced by MRI technology. The temporal resolution, it's a hemodynamic technique, so it's limited to how fast the blood flow can change. So how fast is that? Well, best estimates are about 1 to 1.5 seconds. You'll see, if you imagine it like the brain activity is like a movie, you would see the movie or the picture change about every 1.5 seconds. So that may not seem like a big deal to you, but lots of things that we study are faster than that. So let me give you an example. I'll study recognition memory. So we would have people study a list of words, and then we'd have them take a test where they see a mixed list of old and new items, and then to tell us whether it was an old item or a new item. Well, it takes people on average about 1 to 1.5 seconds to make that judgment. So in 1 or 1.5 seconds, they see a word. So you have visual areas for seeing the word. They identify the word in their semantic database, semantic memory. Then they decide whether or not they've seen the word or not. So you have the memory judgment. Then they have to decide what button to press. Then they actually press the button. So all of that happens within 1 to 1.5 seconds. And all of those areas that I mentioned with those different forms of cognition would be active. And we would have no way of sort of teasing those apart, or let's just say it would be very difficult to tease those apart in that window of time. So this could be a real problem for faster forms of cognition. So something for you to keep in mind with that. And we're also going to see the fMRI, same problem with its temporal resolution is limited by blood flow. Now, the other problem with PET is that you have to scan and block, so it's not event related. And the reason is the way the scan happens. So you inject a label, the label decays, and then creates an opportunity to scan. So you have the scan window, like if you're using O15, you have like a 90-second, maybe a little bit longer scan window where you're getting signal coming from the brain. So during that time, you want the person to engage in all of the same cognition so that you can create the scan. Then you'll have a rest period to make sure that all the radioactive label has decayed. Then you'll repeat the process. You'll have another scan period. So that means that you have to present, like if you were doing recognition, you'd present all old items in one block, one scan, all new items in a different block. And as you might imagine, people can pick up on those sequences and it changes how they do the task. So not very good for those kinds of applications. But if your cognition is uninfluenced by the sequence of trials, then it's perfectly fine. Okay, let's talk about functional magnetic resonance imaging, or FMRI. It's a very common technology that's used to image the brain, and it's based off of magnetic resonance imaging, which uses magnetic fields to create the images. So it's, to give you a sense, the Earth's magnetic field is measured in something called Gauss, and it's about half a Gauss. A Tesla, not the car, but now it's a measurement here, but 1 Tesla is about 10,000 Gauss. So the strong MRIs use roughly 1 to 5 Tesla magnets. I'll show you a video of a 7T magnet or 7 Tesla magnet. Creates a very strong field. So I'm going to show you a couple of videos, or give you access to a couple of videos. I won't do that here because I don't own the rights to these videos, but you can see evidence of how strong a field it is, as well as the quality of the images. The stronger the magnet, the stronger the field. So because it uses a magnetic field, sometimes people forget to remove the metal objects around the scanner and accidents can happen. So, sometimes people die because they fail to secure a propane tank that was in the room, or a floor buffer. And these are floor buffers that got jammed into the bore of an fMRI, and they weigh several hundred pounds. So you can see how strong that magnet is, that it can pull these ferromagnetic objects into it. Obviously, if you have ferromagnetic metal in your body, you can't participate or get into an MRI scanner, so that would screen participants for that before imaging them. So these are some of the videos that I'll link you to that sort of show you. First, it shows you how strong the magnet itself is, and then you'll see an image of a brain using a 3T magnet and an image of a brain using a 7T magnet, so you can see how the clarity of the images gets much better as you have a stronger magnetic field. Okay, so how this works is you have this magnet on, and ordinarily the hydrogen protons are going to be randomly spinning around. But with the magnet on, the hydrogen protons line up. So then the scanners, so they're all, imagine them all lined up perpendicular to the magnetic field. But then the scanner itself emits a radio frequency pulse. So a radio frequency wave, it's part of the electromagnetic spectrum, but we can't detect or feel that. But that radio frequency pulse, this will be referred to as the pulse sequence, causes these protons to bounce, to resonate. And because they bounce, that'll create tiny magnetic fluctuations that are picked up by the scanner, and an image is created. Generally, what will happen is the person will sit into this scanner for several minutes, maybe like 15 or 20 minutes, and they're just doing this pulse sequence, creating a high resolution image of the person's brain. Then they'll start the study itself. And when they start the study itself, they'll take pictures faster. And when they do that, though, they're going to take faster images, and those images aren't as clear as the structural image, right? So you lose a little spatial resolution, but what happens is you overlay that data on the person's structural image a little bit later. Now, the image will be weighted to show hemoglobin in the blood. So this is something called the bold signal. So you'll see this term used a lot, so I want you to know what it means, but it means blood oxygen level dependent. The hemoglobin, so oxygenated hemoglobin, has different magnetic properties than deoxygenated hemoglobin. So that means we could pick up oxygenated blood. And again, it gives us an indication, like the PET scan, gives us an indication of activity by looking at oxygenated blood and where it moves in the brain. So really, fMRI data is a matrix of numbers. So basically, the brain gets divided up into what are called voxels, which are tiny little cubes of tissue. So the voxel will be sort of like 1 millimeter by 1 millimeter by 1 millimeter. And as you can see, the volume is the cube itself. But of course, each cube has activation levels from the bold signal. So hence the name voxels, which combines volume and pixels together. But that information is then overlaid on top of the person's structural image to create the final scan. So here's some MRI images of different participants. So you can see a subject number in the upper left-hand corner of each image. And in each case, the visual areas are active because that's a common sort of thing to have happening in this particular task. So some special software will then do some rendering, and it's going to take the voxel information and overlay it on the structural map. Now, the other thing that happens during signal processing is they overlay it on a brain template. So they kind of stretch each person's brain to fit this common brain template. So that's something else to sort of keep in mind as well. So here's an example. I used this earlier, but this is a fMRI, just a finger tapping task. So you see that motor cortex is active. This gives you a sense of like how many areas are often active in many of our tasks. If you're doing MRI or PET, it's not hard to find areas of significant activation. The issue is, how do we make sense out of these activations? So hopefully you can start to see like the idea behind the subtraction logic is to pare that down, or sometimes we use statistical modeling to decrease or identify areas of interest to narrow down our focus and focus in on key components. Here's an example of fMRI applied to memory. And so sometimes we can differentiate between different kinds of memory, and you'll see that they are overlapped or associated with different areas of activation. So a couple things to keep in mind is there's some noise that can come from the fMRI. So there's some heating noise or power fluctuations. A really big one is head motion effects. If the person doesn't keep their head perfectly still and they move and you're taking images of the brain, well, then that's going to distort your image. So everybody has to correct for head motion in some kind of way. Plus, they'll also, researchers will put their head in a cage to try to minimize head motion as well. Other physiological changes can influence the blood, particularly things that influence blood flow can influence the MRI. Structural differences in tissue, subject cognition can also influence. So timing, using different strategies, engaging in non-task cognition, all of these things could be examples of noise in an fMRI experiment, particularly if you're talking about across different tasks or even across different participants, that they would affect the MRI. So because we're going to talk a lot about MRI studies or read them, I think it's worth explaining a little bit about the signal processing that will happen that you'll read about in the methodology. So people use software. It's very common to use. Some common ones that you'll see is the SPM, or statistical parametric mapping. You'll see Brain Voyager quite commonly. There's a few others. And what they will do is they'll do some slice timing correction to line things up, for example. Head motion correction, I kind of mentioned that before. And then normalizing the brain to a standard brain, also mentioned this as well. And so there's a couple different ways to do that. The MNI is more common now and probably a much better one because it was based on lots of different scans. But essentially what you do is you just take an individual's brain and you stretch it to some model so that you can compare across people. The Talerac-Tourneau was used quite a lot, but it was based on one person, so not a great basis. Then there'll be a lot of spatial filtering or smoothing, and I have an image to sort of show you what that looks like, and so you apply different algorithms. So here's some unsmoothed data, and again, we can smooth this out and sort of identify what areas are a little bit more common across voxel areas, right? And so that kind of narrows down, smooths out some of the noise in our data set. The other thing that is really important to do is to focus on regions of interest and to examine, otherwise it's really exploratory, to examine a predetermined area, how it responds according to the task. So you want to check the papers. Do they mention what areas have been previously shown to be associated with this cognition, and do they zero in on those areas of interest? Now, it could be exploratory research, and if it is exploratory research, they might not have regions of interest, but are there authors explaining that? Do they make that clear? Are their conclusions tempered because it's more exploratory? Those are things that you want to keep in mind. So some strengths for fMRI: it's got great spatial resolution. Remember, it's 1 millimeter voxels there. It's cheaper than PET. Very few risks, just the ferromagnetic thing. Claustrophobia, because it is kind of a claustrophobic environment. It is a little loud, and sometimes people will be wearing, either if they're not wearing headphones, they'll be wearing hearing protection to protect against the loud sounds. The weaknesses, if we're talking about MRI, it's just structural information, but assuming we're talking about fMRI, again, the temporal resolution is bounded by blood flow changes, 1 to 1.5 seconds. So for very fast forms of cognition, this could be an issue. And there are some environmental issues. So I mentioned that like the environment itself being loud, cramped, dark environment can affect behavior. Like remember that Gutchison Park example of how it affected memory. So it's something to keep in mind there as well. But otherwise, it's a great technology which is providing lots of really good evidence. All right, let's talk about event-related potentials, or ERPs. So the way this works is you record EEG, you just place electrodes across the scalp, and you'll time lock to an event, like the presentation of a stimulus, and record essentially the net electrical activity coming from groups of cells deep within the brain. So I have an ERP lab here at TCNJ, and this is the pictures from our basement lab where you place electrodes all across the scalp and record for them. So we'll use, this is a traditional electrode placement. We'll use electrodes all across the scalp there, and you're sampling across the scalp. And this is an example of if we took random segments of EEG and we average them together. Remember the averaging procedure. You can see that there's no signal there. All of its random activity, and it gets canceled out. But with an ERP, if you average around an event, in this case, the sound of a click, you average together to get the ERP, and we can examine this ERP because it tells us about the processing of information in some kind of way. So we can blow this up, and we can look at the peaks and the valleys of the ERP. And in particular, we'll look at how ERPs from two different conditions will relate to one another, look at the difference between the waveforms, and that tells us that it was processed differently in some kind of way. Then we have to make sense out of it depending on what our manipulations were. So let me give an example from the memory field. So if we have people do a recognition test, we can generate an ERP for old items and generate an ERP for new items, or studied and unstudied items. And we can look at the difference here. So this plot is sort of showing you old versus new, as well as some other conditions. And we can look at the individual traces. These represent individual electrodes over time. And you can see that for the first part of this, what's called an epoch or time portion, there's not much of a difference in the conditions, but we see memory-related differences start to emerge, at least in this case, right around 400 milliseconds or so, and it'll continue till about maybe 800 milliseconds or so. Now this topographic plot just plots the old versus new difference across the scalp, so you can see what it's like in terms of the scalp distribution. Where is it largest? So in this case, it's largest at the left parietal area, and that's helpful. What you want to see if you're reading an ERP study, you want to see both the traces and the maps together, because maps could be constructed that kind of distort what's really happening in terms of the latency. You're selecting an interval to graph over, so you want to make sure that there's no latency differences before you start looking at topographic maps and that sort of thing. So there's lots of noise for ERPs. There's bioelectric artifacts. So we're recording EEG, so anything that generates electrical activity could be a source of noise. You have to worry about heart issues if you're not using a head reference, so a cephalic or head reference. Most everyone uses a reference on the head, so that eliminates heart. But skin potentials are an issue. Typically, we reduce electrode impedance, or impedance is resistance in effect. And if you get that really low, like less than 5 kilo ohms or so, you'll reduce the effective skin potentials. You can also use a high input impedance amplifier. That does help, although it's still a little bit noisier than if you use lower impedances, but it can be okay depending on the application. There's lots of environmental artifacts. Anything that produces electricity we could maybe pick up. The other thing to consider is skull thickness. So typically, in a lot of these studies, we're using within subjects design. So the person is serving as their own control. So we're looking at their brain activity in one condition versus another. So yeah, the skull thickness might be an issue for recording EEG, but if you're really comparing the person's ERPs in one condition to ERPs in another condition, that gets held constant. So we typically do that for a lot of these studies just for that reason. So be wary if you're looking at between subjects comparisons, that can be an issue. The skull thickness, the placement of the electrodes, again, is going to vary a little bit from subject to subject. Movement artifacts are a significant source of noise in ERP studies, so you want to minimize movements if at all possible. Now there's some ERP signal processing too that's really important to pay attention to. So we're going to correct for a baseline. So if you look at EEG, the signal moves up and down. So we sample around an event, usually a couple hundred milliseconds before the event is our baseline to sort of zero out and look at the changes from right before when the event was actually presented. We have to remove ocular artifacts because the eyes generate activity as well. There's all kinds of ways to do that, and remove movement artifacts. And of course, we're going to average across conditions. So this is part of the normal signal processing thing. So the strengths for ERPs, very high temporal resolution. We can record as fast as neurons can fire. So we have millisecond timing resolution. So that's fantastic. It's inexpensive, so it's very cheap, relatively cheap. So portable kind of technology. Very few risks for participants. Just minor skin irritation is basically the only risk there. The weaknesses, though, it's a significant one. Very poor spatial resolution. So we can determine that the ERPs differ, but we don't know what brain area is producing that signal. And the reason is because the skull actually blurs the signal itself. So electricity will look for the path of least resistance. So it'll skitter across the scalp, or across the skull rather, until it finds a hole, a suture that it can go out of and into the skin where it's picked up. So that means that it gets much blurrier and less able to identify or pinpoint what structure is active. Now, magnetoencephalogram, very similar to ERP, except so anytime you have an electrical field, which is produced by our neurons being electrically active, you'll also have a magnetic field. It's perpendicular to that electrical field. So we can record the magnetism on the scalp as well. Now, there's an advantage and disadvantage of this. The advantage of it is the MEG signal is less blurred by the skull, so we can pinpoint the structures much better. Now, the downside is to record, you need to use supercooling to record the magnetic signal. So what you're seeing here is a MEG scanner, and there's sensors all along the scalp, just like we would place electrodes, and all of this apparatus here is to do the supercooling to record the magnetic fluctuations. So the strengths of MEG are great temporal resolution, you have better spatial resolution, but it's really not quite on the level of MRI, maybe not even on the level of PET, but a much better spatial resolution. Again, few risks. The problem is it's really expensive. So most of the time when people are gathering the money to build scanners, they're building MRI and maybe doing joint MRI ERP studies rather than spending the money to purchase a MEG scanner. But there are MEG scanners out there. You will see studies, so it's worth talking a little bit about to understand the technology. So a couple of things just to wrap up this discussion about cognitive neuroscience methods. So we've talked a lot about spatial resolution and temporal resolution. Remember, spatial resolution is to identify the structures that are active, and our technologies differ, right? So FMRI has high spatial resolution and ERP has low spatial resolution. So they're going to differ with the information that they can provide. Now, the opposite is actually true of temporal resolution, right? So fMRI has relatively low temporal resolution and ERP has relatively high. So down here, I show you this MRI, the bold signal changing over time for various conditions, and you'll notice that it's on the order of seconds. it also lags behind the actual cognition. That's something you won't see really mentioned in papers, but the bold signal lags behind. So if people make a decision or something, you'll see a change several seconds later. ERPs, on the other hand, are happening instantaneously and on the order of milliseconds. So there's a different activity in time. So the key here is to look at a phenomena using multiple technologies, each capitalizing on their different strengths. That's something I'll talk more about in a second, called convergent validity. So a couple of other issues for you to consider when you're evaluating cognitive neuroscience research. Think about statistical power. So what is statistical power? So it's the ability to find an effect, to reject a null hypothesis when you should. It's a statistical concept, but it's finding a difference when you actually should. So there's things that affect power. So the sample size affects power. So this is an easy way to boost up your power, and used quite commonly in other areas where getting data from individuals is less costly. So if you're in social psychology, you might have really large sample sizes of 200, 300, 400 people. If you're doing correlational studies, might even be more than that. Might be on the magnitude of thousands of people that complete your surveys or give you data in whatever way. So small samples, like that happens with neuroimaging because it's very expensive. So we can't just test a whole bunch of people when it's really, really expensive. So we get sample power, increased sample power, by the number of trials and averages. Remember the averaging. So the averaging also increases power. So the general rule of thumb there is more trials, better power. However, you have to balance that. Remember, these are often within subjects' designs. So if you have a really long research study, people's attention could fade, and that could become an issue. The other thing that affects power is experimental control. So the more things that you can control, the less variability you have in the data. The less variability you have in your data, the greater the power, statistical power. So tighter experimental control can increase power. In fact, this is one of the best ways to increase statistical power, is with tight experimental control. So it's more than just sample size. So it's true, you're gonna want to criticize a study for having a small sample size, but you got to look at the other issues that are involved here. The other thing to consider is, are there confounds in the study? So if it's an experiment, the study will have an independent variable. It's whatever the researcher is manipulating, and typically this is used to explain the observed effect. However, a confound is another variable that varies with the IV that could also explain the observed effect. So you have to sometimes think about that. They may not mention it, but it could also be, but it could be there. And if you have this confounding variable, then you don't know what caused the difference. Is it the IV? Is it this other confounding variable? You don't know, and that compromises the conclusions that you can draw. Now, the key here is, in this field, is drawing or gathering information using a variety of techniques and looking at how the evidence converges on the same kinds of conclusions, something called convergent validity. So what we want to see is, we want to see similar conclusions using a variety of different methods, because these methods have different strengths and weaknesses. So you want to see that. Now, ideally, you would have some patients maybe that provide some information. You have some imaging studies that provide it. Maybe, if you can, you have some animal work that also can inform this. But basically, you're looking for a variety of different methods that are all pointing in the same way. That's the key here, not one individual study. One individual study is going to have some strengths and weaknesses. So this is very important for you to consider as you're reading and becoming a consumer of this scientific literature.